Okay, so I'm going to go over the Succession Lab data analysis. So we're looking at two hypotheses for this lab. The first one is there a difference between the species richness between the different successional stages? And the second one will be, is there a difference between the mean leaf litter depth in the successional stages? So this video I'll split up into two parts. The first part will be hypothesis one, the second part will be hypothesis two. Albeit hypothesis one takes a significantly larger amount of time to complete than the second one does. So I'm going to share my screen. So here we have the data sheet that was uploaded into Canvas. It is just called dataset. And you'll see at the bottom you'll find 4 sheets, one for each of the successional stages. And then I've added here a summary sheet where I like to input data as I'm starting to collate it, just so I can see everything in one place. So for the purposes of this video, I am going to be doing analyses on incomplete data sets. And I'll let you guys figure out what the proper numbers are. It would just take way too much time to go through it all in one video. So I'm just going to be doing the Oldfield as an example for the species richness. So what you'll see here on each of these sheets, you'll have your vegetation survey information on the left, and you'll have your soil and leaf litter data on the right. So starting with the species richness, the first step is really to make a list of all your species and the numbers from each quadrat. And so eventually you're going to have a list that looks like this. To simplify it, what I figured was the easiest thing to do was you'll take this whole table here, and I'm just going to duplicate it so I can mess with it down here. So I'm just going to go through and start deleting a bunch of columns. So after I'm done here, all I will have left are the cells that have data in them. So I'm just going to go ahead and do that really quick. This was just the fastest way that I found to get these lists together. Just gonna delete the rest of this. Okay. So all that we're left with here is this kind of weird looking table. I'm gonna clean it up really quick and just remove all the borders and the fill. So up here, I'm going to go to no borders, and then here, no fill. And I'm also just going to make it unitalicized. And then I'm going to keep everything I have here on the left, and I'm going to add these 2 other columns underneath it. So just copy it, paste it right underneath. Now that that is copied, I'm just going to delete these cells. So highlight them, and then up here on the Home tab, all the way over here under Editing, you'll have this eraser icon, and you're just going to select Clear All. And then the last column I'm going to add underneath. Paste. Okay, and then we do the same thing and just clear these. Okay, so now I have a somewhat concise list. One more thing that I'm going to do is I'm going to delete all the blank cells here so I can squish them all together. If you hold Control every time you select another cell, it'll hold the ones you previously selected. So now we're left with a more concise list. We're going to want to sort all of our species from A to Z. You want to make sure that this is selected, expand the selection, that way that the numbers sort with the species. And now you'll notice that you have a bunch of duplicates that you're going to want to get rid of. But first, we want to sum up all of the occurrences for each of the species. And so to do that, Pinus taeta, loblolly pine only has one, so we'll skip down to Acer rubrum, red maple. It's easily countable right here, it's 5. And so I'm just going to enter it in the top one. And as I go through this list, my assumption is that I'm going to enter the total sum for each species in the first listing, the first row that the species name is listed, and then I'll go back through and delete the second row, third row, and so on. Again, for the second one, here we have 2 columns for this species. And another way to count or easily sum without putting a formula in is to just highlight the 2 cells that you want to sum. And if you look down here at the bottom right-hand corner, you'll have an automatic sum that Excel will calculate for you. So 14, I'm just going to remove the 2 and put 14. And you'll just keep doing this throughout. Again, to sum these 2 up, you just highlight the 2 cells and look at the bottom here for the sum. And then I'll replace that with 402. I'm not going to go through the rest of this list. I've already done it. It is up here. I'm going to go back down. So say that you finish going through this list. I'm just going to go through and delete the extra rows that have the species name multiple times. Didn't mean to do that. Or that. Okay. So control select. And then I would have deleted it if I could actually hold on to it. Delete. And you would do that for the whole thing. Be careful when you're doing this. I've noticed that sometimes the formatting of the names are kind of off. So we have these 2 for examples. Sometimes there's like an extra space in the name, and so if you try to remove the duplicates using this function up here in the home ribbon under data and data tools, you have this function to remove duplicates. And so when I do it here, yes, expand the selection. Okay. Oh, it's not recognizing that there's multiple duplicates here. We're gonna just do it like this. So you'll have situations like this where I went to remove the duplicates, but since there was like a weird space or something is off, it won't actually delete the 2 because it's registering them as 2 unique values. So just double check to make sure that you actually have only a set of unique values here. Okay. So you'll finish doing that, compiling your list of the species and the occurrences, and you're going to do that for each of the successional stages. So now the reason that we're doing this is so we can compile we can complete our species richness ANOVA. And so to do that, we're making an occurrence table, but we need to know the occurrences in a very streamlined fashion. So creating this table makes it a little bit easier. So you completed this. The next step is to create a full species list of any species that you might find in any of the 4 successional stages. And to do this, you're going to do what you just did here by compiling this list. You'll do it for each one and then add them all together. And so once you do that, where is it? It's on here. My examples in the Climax community here. Once you do that, you're going to create a table of occurrences. So here you have a column for species. Let me do this really quick. here you have a column with species, and then you're going to have your occurrences. And what this list is a compilation of all the species from all the successional stages. And then you're going to go through for each successional stage, taking this list, and you're going to mark what occurrences happened within that successional stage. And so over here, this was the list that I made for the climax community. And so I went through, and for each species that occurred here, I listed the occurrence. And then anytime I came across a species that did not occur during the successional stage, I put a 0. And then by the end of that, you'll have a long list, and this is what you're going to be using for your ANOVA. And so what I did at this point was take all of the species, the full species list of occurrences for each successional stage, and summarize them here. This is not accurate with what is in all of these sheets. I used a sample set of species and their occurrences, and I shortened it essentially so I could do this video. These occurrences are not accurate, so you can't actually use them for your report. So what you're going to want to do is go into, let me see. You're going to use the website that I posted on Canvas. It's called Vassar Stats. And this is the website you'll see. There's a video also linked in Canvas that'll show you how to use it. So you want to put your number of samples that you have here in this box. So we have 4, and then you want to make sure you click independent samples. And then you'll copy each one of your successional state data into these 4 columns. So I'm going to do that. Oops. Okay, then we just got the last one pasted in here. And I'm just going to hit calculate down here. It's going to calculate your ANOVA. It'll give you a bunch of different values here, including the standard of error for each one of your 4 samples. So we're going to be looking for your p-value in the ANOVA summary table. Right here on the far right, you have your p-value. So this was insignificant, and so there's no need to do a Tukey's test for this analysis. But if this was to be significant down here in this table, you'll have the breakdown of the significance between every single treatment. So that's that for your ANOVA. You'll want to report your p-value. So this was the aspect of doing this occurrence table like this, was for the purpose of doing an ANOVA. This represents your species richness in a way that we can do a statistical test on it. I'm going to go back here. So for this data point, you're also going to be doing a figure. It'll be a bar graph illustrating the mean species richness for each successional stage. So you're going to need to determine what your species richness is. And to do that, the way this table is set up, you can easily calculate it just using a formula in Excel. So you'll have all your quadrats listed here, and at the bottom of each of these quadrat tables, you'll have 2 total rows. You'll have total number of individuals and total number of new individuals. And you're going to want to look at the total number of new individuals here. And so here we had one total individual. You move the quadrat 2, we have none. 3, we have no new individuals. And so both of these values are 0. And then as you go on, you'll get the new additions of new species. So this is essentially your species richness. And to get that, you can just sum all of these new species, total number new species columns. So I'm just going to do that right now, equals sum. And I'm just going to select each of these. I'm going to ignore these because I don't need the zeros. And there we go. So this is going to be your species richness for the old field. And you're going to do that for all the successional stages and put these values in on your bar graph. So then after that's complete, you're going to want to do the Shannon-Weiner index to look at what species diversity looks like. And so to do this, you're going to take this original list that you just created. So you have your species and all the occurrences for each successional stage. And you're going to be doing the same thing that we did a few weeks ago with the stream ecology lab. So what I'm going to do here is put a bunch of column headings. So the first one will be P sub I. The second one will be P sub I times the natural log. And then the last one is going to be P sub I times the natural log. And then multiply that times P sub I again. And so this is going to give you your H prime value once you sum this last column. So I'm going to just go over it again. To get started, we have our sample size column right here. Just going to label it really quick so you know what it is. Sample size. And so we're going to take the sum of this whole column. And then we're going to use that value to solve for P sub I. P sub I is the species evenness, so it's going to be the sample size divided by 1815, or sorry, it is going to be 1815, but we're going to put it in the formula. And we're going to lock this number in T21 by adding a $sign before the T and then 1 before 21. That way, when we drag our formulas down, it will drag the right value down. And I'm just going to do it one more time. Divide it by this. We're going to add in the $signs and then close out the parentheses and boom. And I'm going to select both of these and then drag and drop the formula down. I'm also going to reduce the number of decimal places. Actually, I'm going to do this again. I expanded it just so I know if it's not 0 or not. Doesn't look like any of them are 0. Okay, so P sub I times the natural log. You're going to type in natural log into the formula, and then multiply it times P sub I. And then again, natural log. And then we're just going to drag and drop this formula all the way down. I'm clicking the last one just to make sure the formula was correct. It is. I forgot down here under the P sub I column, you would want to sum this. And just as a double check here, if everything adds to 1, you've done the math correctly. If it doesn't, you made a mistake somewhere. Then the last column is going to be basically the first 2 columns multiplied times each other. Enter, boom. One more time. I'm just going to drag and drop this formula down. And then to get your H prime value for the old field, you're going to type in, you're going to sum this column, but you're going to want to make sure you take the negative sum of this column. So we're going to put equals, parenthesis, negative sign, parenthesis, sum, select all the values, and then close out all your parentheses. And then that will be your H prime value. Your species richness hypothesis will also have one table, and you can see it in the appendix. Please make it exactly as the formatting shows on there. But you'll want to make sure that you include your H prime values at the very bottom of that table. So that's it for the first hypothesis. It is tedious. It does take a lot more time than the second hypothesis. So the second one is there a difference between mean leaf litter depth in the successional stages? And so on each of your data sheets, on the right-hand side, you have your litter depths, and you're just going to be copying and pasting each of the litter depth, all 10 of the measurements for litter in each successional stage, and then you're going to copy and paste it into your ANOVA. This is my old field. And we're going to grab the mid-transitional. Then we're going to grab the late transitional. And then lastly, let's grab the climax community. And then we're going to hit calculate. Down here at the bottom, we have our p-value, which is significant. And so we know that we're going to go down here and look for the Tukey's test values. And so the Tukey's test value is going to illustrate where the differences lie between all your treatment groups. And so it's going to label all of your treatments as either M1, M2, M3, or M4. And one will be essentially, M1 will represent the data from sample 1, and M2 will represent the data from sample 2, and so on. And so if you want to know the difference between the leaf litter depth between the old field M1 and the mid-transitional forest M2, it'll tell you that your p-value is less than 0.05 and it was significant. And so on, you can get the differences between all of your treatment groups, and you'll want to talk about this in your discussion as well as reporting them in your results. And so that's pretty much the statistical analysis for the second hypothesis. You'll also be creating a bar graph and a table to go along with this data. So the bar graph is going to illustrate the means of each of your leaf litter depths between all the successional stages, including your standard of error. And then your table is going to be a summary of all of the soil data points. And so it's going to be essentially this table right here, but you'll have a row for each successional stage. And so you're going to have one column or one table illustrating all of the data for soil for the different successional stages. That's about it. Let me double check to see if there's anything else I wanted to say. No, I think this is pretty straightforward. There is an extra credit graph, so they're only required graphs for those 2 bar graphs, but if you would like, you may take a stab at the extra credit. Let me know if you have any questions. Otherwise, best of luck writing. Good luck with the rest of the semester.